Swayam Prabha. Digital India. Educated India.
So, we are going a little ahead of what we are supposed to discuss, but I'll tell you the answer for that. So, let me first tell you, now is it clear that from here you have a primary sequence which is basically a polypeptide chain that goes to secondary structure that forms tertiary in this quaternary, right? Now, no matter what you do, so for example, I'll tell you, so if you have a protein and you kind of unfold it means make it to a primary structure, right? It will always try to fold back to its secondary, to its tertiary structure because all the information that is necessary for folding is buried inside the amino acid sequence. Now you also need to give it a proper condition for it to do that, right? Now what do I mean by condition? So do you know what is hydrophobicity and hydrophilicity? What is hydrophobicity? Water repelling any chemical moieties, right? So even in the amino acids, a large number of proteins, for example, all these are called hydrophobic side chains or hydrophobic amino acids. So because he has asked that question, so these are all, you see, alkyl chains, aryl chains, these are all hydrophobic, right? So these are all hydrophobic chains. Now if you leave them in the environment that is suitable for them, they'll again go back to the tertiary structure, final structure. Now, but because they are hydrophobic in nature, now if you leave them in the water as such, so what will happen? So then if there are other hydrophobic chains nearby, they'll try to, not with this chain, but a different protein chain, then they'll try to kind of come together and form as the first one that we discussed, which is aggregate formation, right? So to prevent that aggregate formation, there are enzymes called chaperones that will kind of hold this, okay? That is little advanced for what we are talking now, but so what is important to understand is all the information necessary for folding of a protein is embedded inside the amino acid sequence. Okay, and this was first discovered by, or this was first identified by a person called Christian Anfinsen, for which he got a Nobel Prize. Now, just imagine every time a protein folds back to the same structure, right? That means, suppose I have three variables, right? One is this bond angle, and then there is a dihedral angle. Now, suppose if I have around 60 or 100 amino acid protein, I have a 100 amino acid protein. Now, if I have to sample all the possible conformations and go back to the same state, how much time will it take? Lot of time, but approximate guess on how much time it would take. So I have suppose 101 amino acids, and each one of the amino acid can take three different conformations. So one is this angle can change, this angle can change, and the angle between this, basically the dihedral angle, this plane and this plane can change. So if I have three different possibilities and I have 101 different, so if I have 101 amino acids, how many peptide bonds I will have? If I have 101 amino acids, how many peptide bonds will I have? I'll have 100 peptide bonds. There will be one less, right? So if I have 101 amino acids, I'll have 100 peptide bonds. And if each peptide bond can take three different conformations, how much time it will take to scan all the possible conformations? You know from your combinations, you have done some kind of combinations in mathematics, right? So if I have 100 different moieties and each moiety can take three different conformations, how many different conformations are possible? 3 power 100 kind of conformation. So if I have 101 amino acids which will have 100 peptide bonds, and each peptide bond can have three different conformations. So total conformations that are possible are 3 power 100 conformations. So so if the protein has to scan through all these conformations, because unlike other polymers, so other polymers you don't have this problem because every time you do unfold and leave it, it will take a different shape. So whatever is happening faster kinetically, it will land into it, right? But that is not the case with proteins. The case with proteins is whenever you unfold and leave it, always it goes back to the same conformation. That means it has some kind of already stored information there. So it has to basically scan through 3 per 100 conformations to arrive, if it is scanning all of them, to arrive at the, so what is this conformation called? This is the minimum energy conformation. So it has to scan through so many conformations to get to the final conformation. Can you just guess how much time it will take? 3-4 years. 3-4 years is a way smaller. So I'll tell you how much time do these bond rotations take? What is the time scale of the bond rotation? Huh? These are much smaller, faster than that. Bond rotations happen in picoseconds to femtoseconds. Okay, let's take few tenths of picoseconds, or you can take pico. Bond rotations happen in picoseconds. The vibrations and all those things happen in picoseconds. So each bond rotation can take picoseconds. Now you have so conformations with 3 per 100 conformations. So for 3 per 100 conformation, if each conformation takes picosecond, how much time will it take? I have given you all the information. Just have to do calculation. 3 per 100 conformations are possible, and each conformation can take around a picosecond. Yeah. 35 digit number. More in terms of seconds is much more. Okay, so you can do this math at your home. So it is going to take 10 to the power of 48 years. 10 to the power of 48 years. You can, it's a picosecond. You can multiply with 10. You know what is picosecond? 10 power minus 12, right? You can multiply it with that and then convert that. So basically, I have done a simple calculation here. So it is a 150 amino acids. So 150 amino acids will take around 3 power 150, which is 10 power 68. Okay, conformations. So if each of them is taking a picosecond, it will give rise to 10 power 56 seconds, which is nothing but 10 power 48. This is for 150 amino acids. It is not going to change much for 100 amino acids, a little change. So in principle, if the protein has to fold back, so why this problem is coming is, every time you unfold a protein, it will come back to the same structure. That means it has to scan through the structures, right? So if that, and each structure, even if you take the fastest scanning is only this bond rotation, which is around a picosecond, it is going to take 10 power 48 years. Okay, but do you know how fast a protein folds in reality? It folds in milliseconds to seconds. A protein folds in milliseconds to seconds. That means it is not actually, so isn't it a paradox kind of thing? We are saying that every time the protein has to come to its native state, okay, for that it has to go through either it has to go through it or not go through it. At least it has to go through half of these, right? At least half of this, which is basically giving you 10 power 24, 10 power 48 by 2. And if each bond rotation and other things are taking around picoseconds, technically it should give you 10 power 48 years. But actually the protein folds in a millisecond to few tenths, milliseconds to seconds, okay? Or you can say minute, 1000 seconds, a few minutes, okay? So that means it is happening very fast. How do you think can this is called as Levinthal's paradox. So it's a very famous paradox called Levinthal's paradox, saying that if the protein has to go back to the same structure every time it is unfolded, and it has to scan through all the conformations, this is the time it should take, but it is taking only few milliseconds to seconds. So how is this even possible? Can you guys think of any specific reason why? How can you do this process which should have actually technically taken this long in such a short time scale? How can you do that, or what is the logic that the proteins might be going through this?
So probably that doesn't go through all the possible combinations, just goes to that one combination.
Then because it's a random search, right?
It's programmed in that way to only go to that one possible.
What is defining the program?
The molecular interaction.
The molecular
Interactions, like if one side polar, one side non-polar, they want to come together. So by default.
So basically the energetics of the interactions. Okay, and then
So that all these 100 processes have happened simultaneously.
but it's the same one protein, right? It's not like there are different proteins and all of them are doing, okay, I found the best one. It's not like 100 people are working and saying that, okay, I found the lowest energy. Every protein will go, if you take 30 proteins, unfold it and leave it, all the 30 will go to the same position, right? That means each one of them is working independently, right? No communication between them.
Yes, sir, but there are lots of bonds within the same protein and they could all fold simultaneously.
Okay, that is one thing where basically there is a cooperativity kind of thing where the folding is happening simultaneously. But will it drastically decrease this 10 power 48? Because it has to find out the combination which is having the lowest energy also, right? It's not like one bond is rotating, all of them are rotating and that's the perfect one. All of them have to rotate, keep on changing and find the perfect conformation, right? So there's still a communication has to happen. Not all of them are independent. That takes a locally more stable state. So basically, this is called Levinthal's paradox. As I said, it's a very, very interesting paradox if you take everything to be independent. So a lot of propositions. One proposition is called folding funnel. So this is called folding funnel where. So to start with, you have all so a funnel is basically it will have a bias. Bias means there are certain energy contributions. So it is not going to scan through all the possible angles. It is only going to scan through few possible angles because there are always steric hindrances or steric repulsions between the other one. So it is every time there is basically a configurational entropy. You all know what is entropy, right? Okay, so there is a configurational entropy on the unfolded protein. So it will try to actually, there is an energetic bias that drives a reaction or drives a reaction towards its lowest energy state. So whatever we are saying that this is the lowest, there is only one conformation. That conformation has to be energetically the lowest energy state. Only then always that particular conformation happens. So for something to happen again and again, that has to be the most stable conformation. Most stable conformation means it will have a lowest energy. So this is a huge mathematical model on itself, which we will not be talking now, but it is called, there is a model that has been developed which is called protein folding funnel, where to start with basically the unfolded protein is here, but then it is going to basically, because of the small energetic bias, that means which particular angle is possible, which particular angle is not possible because of the steric hindrance, all that information is there. And then that information will drive it to a lowest energy state, and this is called as protein folding funnel or the energy landscape. And by the way, do you know the person who kind of determined what are the possible angles in a peptide bond and what are the not possible angles in a peptide bond? Do you know the person who has suggested, hypothesized or even proved what are the possible angles that are possible in a peptide bond and what are the angles that are not possible in a peptide bond? So if you can actually know the angles that are possible and not possible, you can predict the structure of the protein, right? So, and you will be surprised to know, and it is very important for you guys to know who is that person. He actually worked in a university very next to this place in Madras University. His name is called G.N. Ramachandran. So it is very, very important for you to know that the biggest problem in understanding the protein energy landscape was actually solved by a person called G.N. Ramachandran, who has developed what is called as the Ramachandran plot. Okay, so he was for a long time working in the physics department of Madras University. Then he moved to IISc Bangalore. He passed away before you were born. He passed away in 2001. Okay, so and he is called as pioneer of this protein structure prediction. So he is unfortunately he is recognized well for this Ramachandran plot, but he is also the one who solved the triple helix structure of collagen. So there is a collagen structure that was solved by him. You can say one of the greatest biophysicists of India or one of the greatest biophysicists the world has ever seen is Professor G.N. Ramachandran. And you should be proud that he did most of his work in this city, okay, in Chennai, when it was called Madras. Okay, so I think whenever you talk about, listen about proteins, whenever you hear about proteins, you should correlate the proteins to protein structures, and a large number of protein structures, the first protein structure or the initial protein structure, collagen, was solved by him. and also he not only just solved one structure, but he gave this entire map called Ramachandran map, which will tell you what are the allowed angles of these two angles. Basically, one is called phi and one is called psi, which will tell you angles that are possible between these two states, and that will result in a protein to fold. Okay, so I think the DST and DBT, all of them commemorate his contributions. So it's important that you guys know him well. Okay, so you read a little bit more about him. So I guess now you all probably know the proteins fold, but you have probably understood how complicated or why is a protein folding interesting problem, right? Why is a protein folding a complicated problem? Why is a protein folding complicated problem?
Because we don't know which way, there are numerous possibilities or 100 possibilities, sir. And we are not exactly certain under which conditions we fall into one of those regions or 100 possibilities.
Right. So basically the information that is there in the amino acid chains is actually one that is driving the proteins to fold. And why is it important to study protein folded structure? That is what is giving the function of the protein, right? Like although there are proteins which have disorder regions that are functional, but for a large number of proteins, the final structural form is a functional form. If you want to develop a drug, you need to know what is the fold of the protein. Only then you can develop a drug. If you want to understand how a protein is functioning, right, you need to understand a structure of the protein. So all these are related to the structure of the protein. So structure of the protein happens because it is folding from a simple polypeptide chain, and that is always conserved. And every time you unfold a protein, you can take it folds back to the same protein or it will aggregate. So that is an interesting thing. For example, there are conditions where you cannot fold back the protein to same structure. For example, you take the egg white, right? Now you heat it, right? It forms that white color blob. What is it? That's an aggregate protein, denatured. So certain times when you heat it or when you unfold it in a positions where it cannot fold back, that means proteins are something like this. Either I fold back to the same protein, a same structure, or if I misfold, I'll create all kind of problems. So that means I have only 1 or 0 kind of answer. You make me fold to the same protein always. If you don't allow me to fold to the same protein, then I'll misfold and create all kinds of problems, right? So that is what the protein philosophy is. So that's why if you see this particular aggregates, right? So these are all because you are not allowing the proteins to fold properly, and that results in this. So that is the interesting part of protein folding. And so there are various models of protein folding. Okay, one of them is called as secondary structure formation. That means first the secondary structure forms, and then they collide and collapse to form a tertiary structure. The other is called as nucleation one, where this is the most important one, which is hydrophobic collapse model. Hydrophobic collapse model means, what are hydrophobic residues? Hydrophobic residues are residues which are supposed to be, they are not supposed to interact with the water. So what they will do is, so they try to avoid the water and they come together. So once they come together, it's like they'll avoid the water, they come together, and that forms the initial nucleation for the protein fold, and then from there the protein starts folding. Okay, so there are various models like hydrophobic collapse model, nucleation model, and secondary structure. These are all various mechanisms of the model. But how do you think we will determine the structure? So you know that the structure the protein has folded. How do you think we will determine the structure of the protein? Or for example, for that matter, how do you know the structure of any molecule? Simulation is still a simulation. Simulation is a prediction, right?
Measure it
NMR.
through NMR or X-ray crystallography.
Through NMR, X-ray crystallography, and there is a much more recent technique called cryo-electron microscope. So these are all techniques. So cryo-electron microscope is nothing but taking the picture of the protein. Okay, it is like just like you're capturing a photo of the protein. And also, what do you think will be the size of this folded protein? We have been talking and we have been writing it. Can you guess or estimate the size of these proteins? 10 power minus 15 to 10 power minus 20. That means femto to less than attometers. Okay, let me tell you. What is the size of a hydrogen atom? 10 per 10 per minus 10 meters. So I guess you know that the radius of the hydrogen from nucleus to the electron, what is it? You must have studied it. It's a 0.529 angstroms, right? The distance between the nucleus and the hydrogen atom is, the radius is 0.529. That means the entire diameter of a hydrogen atom would be double this, right? It is around 1 angstrom. 1 angstrom is 10 power minus 10 meters, right? So 10 power minus 10 meters is hydrogen atom. So this cannot be correct, right? So, and we have a large number of structures that are forming. So hydrogen is the smallest. We have a carbon, oxygen, and then you have a huge number of structures. So what did
Micrometers, milli to micrometers.
micrometers? So if it is a millimeter, you can actually see. Our naked eye can see millimeter. So can I see my proteins? No. Micrometers can also be seen easily by microscope. A simple microscope, if you take, you can see a micro. Then why do you have to use so much complicated method like NMR, X-ray, then Cryo-EM? All those are not needed. You put it in a microscope, you'll get the structure. Nanometer. So these are all in this. For example, this might be around 2 to 3 nanometers. So depending on, so it will be between anywhere between 2 to 20 nanometers, depending on how big the protein is. Okay. So we are talking about entities. So that is why these are called as nanomotors or nanomachines. Proteins are called nanomotors or nanomachines because they are like machines or motors at a nanometer scale and acting as this motors or machines or whatever you want to call. So we have to determine the structure of these proteins, which are nanomachines or nanomotors. And so as somebody said, there are multiple methods. Okay, by the way, you asked me about chaperone. So let me tell you a very quick thing about chaperone as I discussed. So if you look at the protein folding, right? Now these are various models. The information regarding the fold of a protein is inbuilt in its amino acid sequence. But only when you are not allowing it to interact with the adjacent proteins. Then if you have a hydrophobic residue, instead of burying itself, it will start interacting with the other hydrophobic patches, right? So then they'll all come together and form aggregates. So in order to prevent this interaction of one protein with the other protein molecule, for few cases, there are these enzymes called chaperones. Okay, these are like, some kind of boxes where these are proteins. So these are basically chaperones where the protein enters into it, the unfolded protein enters into it. So that unfolded protein is no longer interacting with other unfolded proteins, and then it kind of folds and gives you a folded protein. So these are kind of boxes or it kind of enzymes where they're not really enzymes, but they're called chaperones where they'll try to chaperone the interaction of one protein with the other protein so that they kind of fold to a same simple structure. Okay. Now, as I said, there are various ways of determining structures: X-ray crystallography, nuclear NMR, Cryo-EM, and all those. So these are all. So, for example, I don't know if you have a tour to Cryo-EM center. We have a Cryo-EM machine in our institute. So, is it there in your schedule? Visit to Cryo-EM center? No. Crystallography depends on the diffraction, right? So basically you have crystals of it. Now crystals have a repetitive patterns of the same unit, right? Once they have a repetitive patterns, they'll diffract the light. So that will give you a specific pattern that is called crystallography. So we have a single crystal XRD in the ground floor of this building. Yes. So, but that is not powerful enough to give the structures of the proteins. It will give you structures of small molecules, but still the principle is the same. You must have heard of Bragg's law? Probably not. Okay, fine.
Yes, sir.
What is Bragg's law?
Something. So it's used in quantum mechanics where if you rotate something two-dimensionally or three-dimensionally, if you reflect it, won't come back to the same space, I believe. So I might be wrong.
Okay, so Bragg's law is slightly different. Bragg's law is telling you the wavelength, so basically the wavelength of the light that is diffracted depends on the spacing of it. Basically there is something called n lambda is equals to 2d sine theta, where the spacing of the units will tell you the wavelength of the light that kind of interacts with that. Okay, so but let's not go ahead of it, but you can see the single crystal XRD here, you can see NMR, you can see cryo. All these three instruments are there in IIT Madras. In this building, these two are there. So you can take a look, and these are the machines that give you the structures of the proteins. Okay, so now we have talked about for a long time, more than 1.5 hours or close to 1.5 hours, we talked about the folding of the protein, right? Now what happens if the protein doesn't fold properly? Oh, okay. There you can also, by the way, one quick thing is you can also, this is experimental methods, but you can also computationally predict the structures of the protein. And as we discussed in the beginning, computationally predicting the structures of the protein was awarded Nobel Prize in 2024. So it is basically called AlphaFold. So if you guys are interested, you should go and download or work on what is called AlphaFold. Or there is another game kind of thing which is called Fold It. So Fold It is actually a game, it's basically a game which allows you to fold the proteins into its lowest energy conformation. Fold it. So if you have time and if you have interest, you should go and explore these AlphaFold and Fold It kind of softwares. Yes.
It's this is slightly out of topic, but then we say in AlphaFold 3, which was making the rounds in the news a few months ago, we say that the model has not only gained the capability to predict protein structures, we also talk about RNA structures, right? So, would you like
So AlphaFold, initially to start with, it was designed to develop the protein folding structures, so structures of the proteins. Then it was basically as it kept on increasing its power. So people were able to study protein-protein interaction. We did not talk about protein-protein interactions, but I think in the initial PS slide I have showed you, or one of the slides, how a spike protein is interacting with ACE2 protein. That is nothing but an interaction of protein with other protein. And you know, none of these protein interactions are covalent linkages. All of them are non-covalent interactions, right? So AlphaFold is able to predict protein-protein interactions. These days, protein nucleic acid interactions. And RNA or any other nucleic acid structures are actually much easier to predict. So I'm sure AlphaFold will be easy. It will be easily, because the complexity of nucleic acid is only 2. There are only 4 different bases, right? So you can always predict how these 4 bases are interacting. Whereas amino acids, there are 20 different amino acids. So how the interaction between these 20 different amino acids is much more complex than the bases. So
proteins and RNA work together form a new structure.
yes, so that those are called protein nucleic acid complexes that AlphaFold is able to predict. Okay, so you should go and read more about it. So I think not just for this class, but it is always a, so how many of you know of a website that publishes the Nobel Prize information? Nobelprize.org. So every year, you know that a Nobel Prize is awarded for the best possible scientific discovery or invention, right? So there is always a 1 or 2 page article on every Nobel Prize winning accomplishment in for a layman terms. That means layman means not going very technically, but only simple words. So anytime you get chance, you should go to that website and you should start reading why was this discovery important, or why was this invention important, and how did they do it. So you should also understand how did they do it. Then only you will be able to appreciate it, right? So you can start doing it from this year or any time. Whenever Nobel Prize is announced, or even the prior years, you go to this website called nobelprize.org, and it will give you all the information. So it will give you who are the scientists, and it will give you all the information on why was it awarded, why is it important, and how did they arrive at that particular conclusion. Okay, that will give you, that will help you in understanding how the science progresses. Okay, so this is about protein folding. Now let's move on to protein misfolding for the next 10 minutes. Okay, so you have, as I said, you have a folding that is there, but this folded structure need not always come back to the same structure. If suppose sometimes the protein folding goes wrong, it will give rise to what is called as this misfolded proteins. Okay, sometimes misfolded proteins are much more stronger and much more energetic because it's not a simple fold. It is interaction between various folded proteins or various misfolded proteins, and so it can actually, they are much more stable. Many of the misfolded proteins are once they form other quaternary structures, they are much more stable, and that will result in a large number of diseases like you name any of the neurodegenerative disease, for example, or even other diseases, the primary cause is a protein misfolding. For example, Parkinson's. You know what is Parkinson's, right? It's also a neurodegenerative disease. And do you know what happens in Parkinson's? Parkinson's happens because there's a protein called alpha-synuclein. Okay, alpha-synuclein protein misfolds, and that results in Parkinson's. And Alzheimer's, as I said, that is because of a misfolding of amyloid beta. And there is another famous common disease that you generally see in bovines and other things. It's called mad cow disease. Okay. Yeah, it is mad cow disease. It is because of a aggregation of or misfolding of a protein called tau protein. Okay, so you take a large number of diseases that are actually caused because proteins do not fold to its correct structure. Now there are two things. Protein do not fold to its correct structure. So because of that, what happens? The function is lost, right? Because we know that the function of the protein is largely determined by the structure of the protein. Now, if the protein doesn't fold back to its original structure, the function is lost. So that will result in some diseases. There are other diseases where if the protein doesn't fold back to its native structure, that's called misfolded structure, and these misfolded structures come together, and that will result in a other kind of set of diseases, mostly plaques. They create a lot of plaques and they kind of fibrils in your body, and that will result in diseases like neurodegenerative diseases, Parkinson's and Alzheimer's and other kind of things. Okay, so that is why. So if I showed you a protein folding funnel, right? A protein folding funnel is something like this, where you have a native low state. But this, if it goes to an intermediate state, if it goes to a misfolded state, it can form a large number of aggregates, and that aggregation will lead to a more stable. You can see that this is an energy axis. So this is the energy axis. This is at a much lower energy than this. So this are a lower energy forms. So lower energy means more stable forms. So that's why it's not easy to get rid of these aggregates, right? Okay, there are three different kinds of aggregates. One is amyloid aggregates, inclusion bodies, and soluble oligomers. Amyloid aggregates means, so basically you have a fiber, you have a protein that is misfolded, and it will form first a dimer, it will form a tetramer, it will form a octamer, it will form large fibers, and once it forms large fibers, this is a monomer that forms a protofilament. Protofilament means more misfolded proteins will come together. It will form large number of protofilaments. It will form amyloid beta fibrils, and that will result in plaques. So these plaques are the ones that will. So as somebody asked me, why don't if the protein folding is a problem, why don't we see the disease in the beginning? Because it starts with this stage, and it takes a long time to progress to this stage. So you will not see symptoms during any of these stages. You will only see symptoms during this stage. So that is why any of the early interventions, like if you can have early diagnostics where you could detect at this stage, then and you don't allow the disease to progress to this stage, then you can avoid it. Once this particular stage happens, it will not be possible to avoid it because there are enough number of plaques that will kind of block your neuronal activity. And there are something called inclusion bodies. So as I said, these are fibrils, right? Misfolded structures. They are something called inclusion bodies where misfolded structures, they'll not form fibrils or highly ordered structures, but they'll form like amorphous aggregates. So you know what are amorphous in nature, in the sense all the misfolded regions come together and they form what is called as inclusion bodies. So there are various kinds of inclusion bodies that are there, and then you don't worry about the mechanism of aggregation. So as I said, so these are some of the diseases that you generally see because of protein aggregation. As I said, Parkinson's, Alzheimer's, then type 2 diabetes, Huntington's disease. Huntington's disease is another commonly found disease because there is poly-Q amino acids that are largely formed, and then these amino acids come together and form Huntington's disease. There's ALS and other prion disease, which is basically a case of tau protein aggregation. and all this. A lot of diseases that are caused because your proteins are not folded correctly and they kind of form aggregates. Now, so protein folding is important, protein folding is not important. But does our cell have any machinery to avoid this? You should remember that not just human beings, any biological cell or any biological machinery has a large number of proof checking. So it is not like you give me something. So there are like hundreds of proof checkers on the way. Okay. So even in humans or even in living systems, there is a huge proof checking mechanism, and this entire process of maintaining a healthy proteome is called proteostasis network. Okay. Okay, these are all examples of how polyQ forms and how neurodegeneration happens and other kind of things. Okay, so let's not. Okay, so a large number of proteins, as I said, when you have a misfolding protein, it's not like any misfolded protein is directly they'll start aggregating and form this amyloid beta and other kind of amyloids and plaques. There is also a machinery which is called degradation machinery and disaggregation machinery. There are enzymes that degrade specifically misfolded proteins, and there are enzymes that specifically degrade or disaggregate these aggregated proteins. So these are all they're all part of your protein control mechanism or proteasomal machinery, which actually helps in degrading or disaggregating it. Okay, so we will not talk about that. So because it's a little more in detail. So what I'll do is I'll talk about a simple machine called, okay, so I'll only talk about this machine, which is basically a protein degradation machinery, and then I'll stop. Okay, so if you have a misfolded protein or if you have a proteins that are not folded properly, so those folded, as I said, there are molecular motors which actually use this energy. So basically, if suppose this is a protein that has not been folded properly, then what happens is in your cell, there is a machinery that chemically tags a specific peptide or protein to it, and this peptide or protein is recognized by these enzymes. These enzymes are called proteases or proteosomes. So if you see a human body, we have a lot of proteosomes. Proteosomes means, till now we are only talking about protein synthesis, protein folding, protein misfolding. But anything that has been produced has to be at the end of the day degraded also, right? So this is a cycle. So you have a synthesis of the protein, and synthesis of protein is done by what enzymes? Synthesis of protein is done by ribosomes, right? So this is the entire cycle. So you have proteins that are synthesized by ribosomes, then they function, and then if they don't function properly, they aggregate. And if not, finally at the end of the day, these proteins have to be again regenerated into their amino acids, right? This cycle has to continue. And these enzymes that degrade these proteins or break down these proteins are called as proteases, okay, or proteosomes. These have this unique machinery. They are like your molecular motors. So they are very, very interesting enzymes. So you have these enzymes which are called proteases, where one component is only to recognize and unfold. Okay, you cannot degrade a protein that is folded. You have to first unfold a protein. So you basically have this protein which recognizes the misfolded protein or anything. It tries to unfold, and then it translocates into this purple one, which is basically a peptide. It does the activity of this peptide bond hydrolysis. So once it does the peptide bond hydrolysis, then it will basically give you the individual amino acids. So these enzymes that actually do this, these are called mechanoenzymes because they are continuously using the power of this ATP hydrolysis. They convert the ATP chemical energy into mechanical energy to pull on these substrates and then unfold them and degrade them. So don't think that if a protein is misfolded, then it will result in a huge diseases. If a protein is misfolded, if it forms aggregates, then we have enzymes called disaggregases that kind of disaggregate the enzyme, or there are enzymes called proteases and proteosomes that degrade these proteins, a misfolded protein or even a properly folded protein after its life is over. It will degrade it and then it will regenerate these amino acids. So the entire life cycle of the protein is completed. So you have a synthesis of the protein, you have a folding of a protein, you have a function of the protein, and if the folding doesn't happen, it will aggregate and it might create a disease. But there are machineries which can disaggregate, and after the function of the protein, it has to be degraded, and that is done by these enzymes called proteases. And again, these proteases are nothing but again proteins. Okay, so this will degrade them into amino acids. So this is the full life cycle of proteins that we basically discussed in the last 1 hour 45 minutes. So this is about a life history of proteins. So how proteins are synthesized and how they are useful in your machinery, and why is it important to understand the proteins. Okay, so I'll end my talk here.
Swayam Prabha. Digital India. Educated India.